And welcome indeed to the lecture on regulatory science and translational research. If the technology works well, you should see my PowerPoint presentation. If not, please shout.
We see it.
Indeed, I will not go through much of what I did before. You said it nicely, Ulrich. Just to say that I'm now located in the north of Denmark, so not right about around the corner, but even further north than you are. Um, this is as the start of the lecture series now again, and the part one is, why is knowledge in regulatory science important for translational medicine and basic science? So any researcher, why should they actually care? Why should they care about the regulatory agencies? And I will touch upon the role of the regulatory agencies in translational medicine development, but also about medicinal product development in research projects. We don't even know that you are actually working on this. And then about non-clinical science, which is basically what translational medicine's development is. So you have a concept, you want to make this into a medicinal product or a medical purpose, and that is one day a medicine. And this process is called translation. So you test it in animals, you do proof of concept in animal models. Many researchers obviously do that. Now let's start very briefly. What is regulatory science actually? There's many definitions out there. One of them, which I like a lot, is from Fitzgerald. Regulatory science may be defined as the acquisition and analysis of data sufficient to inform decision-making, pertinent to the approval and monitoring of safe and effective therapeutics. So in other words, how can we define the benefits and the risks of medicines on a scientific basis? Is this an arbitrary decision? Something works and it's kind of safe, so let's approve it? Or is there more to this? And how can we basically get there? And how can we define the road towards that? And there's a number of papers out there which I would think are regulatory science papers. For example, analyzing, that was from the European Medicines Agency some years ago, what factors actually influence the approval or non-approval of drugs in Europe? Is there a system behind this, or a systematic things that you could think about? And the one on the bottom right actually was a very interesting one. Now we have had a pandemic, unfortunately, the coronavirus pandemic, but at the time of the so-called swine flu pandemic, the H1N1 pandemic in 2009, after this was over, then colleagues there analyzed with statistical measures the risk-benefit impact of the decision-making for the H1N1 influenza vaccines. If this would have been a more severe pandemic, which at the time one couldn't know, how much kind of cases of death or severe disease would happen if the committee that at the time, of course now as well, is responsible for making these approval decisions, would have come to a decision a month later. How much would that have impacted? And it would have impacted a lot. And that, of course, is important knowledge to have. Now, we're talking very often about Moore's law when we talk about microprocessors. It's now a bit outdated, Moore's law. The number of transistors would double every year. But of course, we do see an evolution in our computers and mobile phones. Look at your mobile phone and compare it with what you had 10 years ago. That's quite a difference. And this is what we call Moore's law. And actually, in pharmaceutical development, this is exactly the opposite. There is less and less drugs, at least it was so some time. And people call this Irum's law, which is Moore's law backwards. And that, of course, was a big wake-up call also for regulators, because more and more requirements would not necessarily help medicine's development. And if you look at graphics like this, which I'm sure you have seen before, of 10,000 substances in research and development, only one is going to be approved at the end, because there is so many critical steps. Like some of them just do not make it into the preclinical phase because they are toxic, or the pharmacokinetics are not suitable. Think about an antibiotic with a half-life of 1 minute. That wouldn't really help anything. And then it goes to clinical development, and that is a cumbersome approach. If you look at the bottom, you can see how many years these are the years, how many years this takes. The whole value process can take 20 years. And that, of course, is all too long. If we have an idea today, we cannot have an approval of this in 2044. We have to be much faster than this. And regulatory science scrutinizes what we do as regulators and as developers and tries to make this better. And very often you hear about regulatory affairs. Regulatory affairs is basically also an important discipline. This is about how to comply with regulations, how to submit a file to the regulators. It's more reactive. It's based on the integration of the regulation. So how do we comply with the blood products regulation, with the human drug law? How do we apply with genetically modified organism regulation, and so on and so forth? And then sometimes strives to fit science into the regulation. So what do we have to do in order to fit the regulation? But sometimes that would not work. And this is what regulatory science basically is, how to evolve and optimize regulations. It's more proactive. It's based on the analysis of regulations to say, well, this is what the regulations are. Where do we have to adapt them? It then strives to fit the regulation into science and update that. And that has actually become a big theme for the regulators. The European Medicines Agency has a regulatory science strategy, and so does the US Food and Drug Administration, US FDA. So this is really a timely topic as well for a lecture series. Now let's look at basic science projects to see if there is any pharmaceutical or medicinal product development there. Let's first define what a medicine actually is. And this sounds more complicated than it actually is. If you want to memorize one directive, one law, then it would be this one, Directive 2001/83 as amended. Sounds very old. It's from 2001. It is actually very old, but it has been amended all the way through. This is why it's called as amended. And it is basically the so-called human codex. It is the drug law that also has been translated into the German Arzneimittelgesetz, the German drug law. So the annex defining the requirements is identical with the one that we have in Germany, in Denmark, and other places as well. And a medicine is defined as how it is presented. So any substance or combination of substances presented as having properties for treating or preventing disease in human beings, or any substance or combination of substances which may be used in or administered to human beings, either with a view to restoring, correcting, or modifying physiological functions by exerting a pharmacological, immunological, or metabolic action. It took a long time to read this. It is a bit complicated, or making a medical diagnosis. So basically, it is about the intention and the presentation. So yogurt, for example, that you eat every morning, would also be modifying physiological functions, but it is not given with a view to do this, and it is not presented in treating a disease. If someone was to take yogurt and write on the package, this is for treating Crohn's disease, then it would be a medicine. And you can see that there's a lot of borderline situations where something may or may not be a medicine, something may or may not be a cosmetic, for example, and something may or may not be a medical device, and so on. So the regulatory agencies do have own divisions who are only there to view borderline situations and make a decision, is something a medicine or not. And as an example, this is from my own research when I was a young postdoc. Ulrich mentioned it. That is quite some time ago. The working group I worked in, Martinsried, actually had a very interesting case of one patient with an autoimmune disease, polymyositis, which is an inflammation of the muscles, where autoreactive T cells would attack and destroy muscle fibers. And normally this is alpha beta receptor T cells. In this particular one case, this was a gamma delta T cell receptor, and this was a monoclonal gamma delta T cell receptor. So you can see on the immunohistology on the right-hand side, this was almost exclusively attacked. These are the muscle fibers, these big round lumps there. And the yellow bit are the gamma delta T cell receptors. And gamma delta T cell receptors recognize the antigen not not within the MHC context, but almost like you could say an antibody. And the idea was, if you could clone this T cell receptor and find what antigen it binds, you could not only define what the antigen is and find out about what causes the disease, maybe also in other patients. Maybe this is just a fortunate case where one can find out what the antigen is for all polymyositis cases, but also maybe use this antigen to induce tolerance against this. And this is where what they worked on. And actually, after I left, they characterized this more. And in 2012, they actually found the target specificity of this. If I remember correctly, it's a certain enzyme moiety that it binds against. And I'm saying this because when you have that idea, you could use it therapeutically or diagnostically in a patient. This is basically early medicines development. And if you would not document everything well and characterize everything well, you would miss a trick, because later on you might need exactly those data. And that is what I'm talking about. So having a lab book and making good notes, even of failed experiments, is a good thing to do, because you have got all the documentation that you may need, or that people taking over at a later time actually may need. And we do also have borderline cases, as I mentioned before, which are not so easy. And this is also where the regulatory agency can come in. Like, for example, this machine, it is easily said, it is a machine where you put fatty tissue in on one end and you get stem cells out on the other side. There's enzymatic digestion in it, and it's a complex machine, as you can see. But according to the definition, these stem cells would be substantially manipulated, as we call them, because they've been enzymatically digested. And they would fall in Europe under the definition of an advanced therapy medicinal product. But if you use this in the operations theater with the same patient in the same session, how could the European Medicines Agency regulate this? And what is actually the product? Is it the medical device making these, or is it the product that comes out of the medical device? Is this the manufacturing process, or is it a medical device that you're using? So this is a borderline situation, and you can go either way. Now, let's then look a bit more at the role of the regulatory agency in translational medicine. And I've used again the advanced therapies that I mentioned as paradigm cases. Advanced therapies are defined as gene therapy medicinal products, cell-based medicinal products, and tissue engineering products. And we do have two agencies in Germany, the Paul-Ehrlich Institute in Langen, close to Frankfurt, which is for biomedicines and vaccines. So, for example, advanced therapies are regulated by the Paul-Ehrlich Institute, and blood products and everything that comes from it, and vaccines, as I said. And then there is the BfArM, the Bundesinstitut für Arzneimittel und Medizinprodukte in Bonn, BfArM, which is the larger agency for all other medicines, and it includes also some biologics. And what we have seen with advanced therapies a lot is that there is top science, top basic research, very complex products. And then we've got these directives and regulations. And basically the directives and regulations also decided that the marketing authorization would not be national. So the Paul-Ehrlich Institute would not approve a medicine only for a gene therapy only for Germany. No, it is mandatory for the centralized procedure. So it's via the European Medicines Agency in Amsterdam, formerly in London, now in Amsterdam after Brexit. And that is quite a gap, because an academic group top in basic science, publishing in Nature and other top journals, would have a long road to travel if there was no help to get something into a medicine. And I'm not only talking about funding, but also all the requirements that are actually there. And at the time, we wanted to also convince the other regulators and create more awareness that this is actually not trivial. And some colleagues looked into the HudraCT database. That is an outdated paper now, of course, but it's still valid in its message. They analyzed 318 trials with advanced therapies and looked at actually who is the sponsor. And normally it would be big pharma, maybe some small pharma. And here you can see it in orange on the right-hand side that 60% are non-commercial sponsors, 50% academia, and 10% charities. So for rare diseases, some charities have picked up the chance and said, we need to do something. We need to sponsor trials for this promising gene therapy in XYZ disease because nobody else would do it. And only 40% were commercial sponsors. And of these, if you break it up, only 4% were actually large pharmaceutical companies. The rest were small and medium-sized enterprises, SMEs, so small companies. It doesn't matter in this case if they are registered or not. You can see that 96% is small companies. And now in my work with clients, with companies, I still see the same. Many companies are small startup companies with one asset, with one gene therapy, for example. And obviously, we do still have that translational gap, and regulatory science obviously helps them as well. So for example, what do we have to do? What kind of animal testing do we have to do? Our medicine does not work in any species other than humans. What are we going to do? I'll come back to this a bit later again. And this is why the regulators have created some soft landing zones, as they call it. The European Medicines Agency has something called Innovation Task Force. The Innovation Task Force is a group of people at the European Medicines Agency. I should say the European Medicines Agency is doing the overarching organization of medicines assessment and development. The member states do send the delegates to the committees, and the committees make the decisions. So there's a German delegate, is a Danish delegate, and so on. But obviously, that Innovation Task Force is organized by the EMA with the possibility of also of committee and working party members from the various groups to participate. And that can be a very powerful type of meeting. It is not very formal. Groups can come. I also saw academic groups actually traveling there and presenting their concept and get a first impression on what the regulators think. And actually also the the German Paul-Ehrlich Institute and the BfArM, the agencies also have innovation offices now. I think most large agencies have that, where people can just ring and say, look, we've got this early development here. We're not quite sure what to do. Can we come and present and maybe discuss the first steps towards a clinical trial authorization, for example? And the regulators will not send them away usually and say, well, come in 5 years when you have 500 pages more of data. But they will say, let's look at the concept, what you are proposing, and see if it is compliant with what we want. And that is actually very good. So the role of the regulator has changed to not only be gatekeepers, but also enablers of development. And when you look at the Innovation Task Force, EMA has published the 2022 report. The 2023 report is not published yet. I think it's coming out in May normally. Then you can see that most clients, you could say, coming to EMA is indeed small companies and also academia. But it's interesting to see that also medium and large pharmaceutical companies actually have taken that on board. And maybe one of the reasons is that there's so many new technologies. If you look on the right-hand side on what enabling technologies were discussed, 3D printing, I'm not reading the whole list, but 3D printing organoids, in silico models, mobile or portable manufacturing. There's a lot of cutting edge technologies there, which obviously makes sense to be tested a bit with the regulator before becoming more formal in a so-called scientific advice procedure. And the European Commission at the time said for advanced therapies, we need to have incentives. And one of them is the certification procedure. And the certification procedure is open to small and medium-sized enterprises. And I should say that becoming registered as a small and medium-sized enterprise as an academic spin-off is not difficult. The EMA has an SME office. with very nice people sitting at the other end of the phone, and this is actually possible to become registered, and then one gets a lot of incentives, and one of that is this certification procedure. It's a bit of a complicated text here. That is the copy paste from the regulation. Basically, what it says is that the pharmaceutical quality, so the characterization of the drug substance, how it is presented, how sterile it is, and all these things, all these data, and also when available, the non-clinical data, so that is the proof of concept data in animals, toxicology data, assays, functional assays, and so on, can be evaluated and certified if they are in accordance with the relevant annex of the of the directive. And obviously, in the early days, it was a bit difficult for the assessors and the agencies. Are we actually assessing this from a marketing authorization perspective? Then, of course, you have 500 problems with an early development because it's not mature yet. But of course, it is stage specific. And there was also procedural guidance at that time. It still exists. It says it's not binding for future marketing authorization. It's not scientific advice. It's not binding for national agencies for clinical trial authorization. So of course people say, well, what is it then? What's the point of it? And the point of this is that it's the only data assessment procedure before marketing authorization application. So basically the only time when a regulator really looks into data and assesses the data. All other interactions are only advice. So for example, asking, is the design of our clinical trial okay? And then the regulator says yes or no, but it doesn't assess data. And in this case, it does and gives a certain benchmark. And my interpretation for what this actually is this. I'm sure all of you or most of you will have done this, a PCR reaction, and you put your products, your tests onto an agarose gel, and then you get this after electrophoresis. And what does this mean? It doesn't tell you anything unless you have a standard, a positive control, and a negative control. If you have that, then of course it makes sense. And for medicines development, if it's the only product, if it's the first product, you have no clue where you stand unless you get a benchmark. And that is what certification actually does. It will come with a lot of different advice and issues that you need to do this and this and this and that, but then at least one knows that. And the idea behind this was that this could then help to have a stamp and get new money in, new funding in. The other incentive where regulatory agencies need a lot of regulatory science is classification. And in this regulation for advanced therapies, it basically says that any applicant developing a product based on gene, cells, or tissues can ask the European Medicines Agency for a scientific classification. Is it an advanced therapy medicine product or not? You may ask, why should I care? Well, if something is an advanced therapy medicine product, you not only know what requirements there are, but also there are certain incentives, indeed, like the certification procedure, like involvement of the Committee for Advanced Therapies, which is the best experts available in the regulatory agencies to input into advice and so on. So there's a lot coming with this. I think there's also some incentives with fees and so on. So it is worthwhile going through this because it gives regulatory certainty. And at the time, I started with this and the committee work started at the time. We thought, well, it's actually easy. What's the point of this? If something contains genes, it's a gene therapy. If it's cells, it's a cell therapy. And if it's tissue, it's tissue engineering. So what's the problem? And actually, if you look at the details, it actually is not so straightforward. So for example, bacteria, you take bacteria like yogurt bacteria, lactobacillus, you take them, you genetically modify them to express on their surface a certain factor which is anti-inflammatory. Then you give it to patients to eat who have a severe inflammation in the mouth. Is this a gene therapy? Because basically it's genetically modified bacteria, but it does not change the human genome. It doesn't, because and even anatomically seen, the oral cavity is the outside world. So you don't even add it into the human. But on the other hand, you add a genetic sequence to the human because they ingest it. And there were long discussions at the time, and the commission then gave us the hint to say, well, it is as it is written. So basically, this is a gene therapy with all the requirements coming with it. Another example is, and this is why the Committee for Advanced Therapies also at the time created a reflection paper where they put their thinking, when something is substantially manipulated, it would be an advanced therapy medicinal product. And this is important, for example, because there is transplantation product. So you take bone marrow from a donor and you give it to a patient, for example, with cancer who has been irradiated, and you do a stem cell transplantation. Is this a cell-based medicinal product which has to be regulated by the European Medicines Agency? That would have been a catastrophe because that's medical practice for decades. Why would all these requirements all for a sudden be valid for something that's already tested? And this is why the legislator at the time came up to say anything that's substantially manipulated. And the committee at the time said cell culturing leading to expansion is considered substantial manipulation, because you don't know when you put it into the hood and have it overnight there if this may modify the cells, at least primary cultures of cells. Of course, for a researcher, that is an alarm bell, because what you put into the hood overnight and have as a cell line should never change. Otherwise, no experiment would ever be believed again. So you see that there is actually quite a bit of conservatism in this, maybe for a good reason. But then how about things like this? This was one product, which is lymphocytes, which you could use by leukapheresis. So, for example, from PBMCs, and you would incubate them with viral peptides, for example, so for certain antigens. And you do this by cytokine capture. So these magnetic beads, for example, magnetic beads, an antibody against interferon gamma on the surface of the cells, you would take them out by a Clinimax system. You would have them incubated, and then you give them into patients. Is this already cell culture? When does cell culture start and incubation end? So anything above 3 hours or 2 hours or overnight? What if you're late in the night and early in the, you see what I mean? This is completely arbitrary. And this is transient activation, and it's basically what T cells are supposed to do. If you put an antigen in the right context to a T cell, it will be activated. Even in my body now, if it sees a flu virus, it will of course be activated and then it goes back. This doesn't mean that I'm substantially manipulated all for a sudden. So there were long discussions around this, but then at the end of the day, the majority decided that this is actually a somatic cell therapy medicinal product. And that of course rose a lot of questions. If it would not be substantial manipulation, how can we be sure? So what about safety, certain alterations, and how, on the other hand, do you avoid that virtually anything is advanced therapies? Because you could say for anything you do, you could say, well, it could actually have altered the cell to a certain extent. We took it out of the body, it stayed out a bit for too long, we centrifuged it. That might actually do something to the cells. And this is why also in this, and regulatory scientists inputted into this at the time it was written, that centrifugation, for example, is not substantial manipulation. And at the time, also pragmatically, it was decided that these more old-fashioned cases like antibody-mediated purification that is used every day in hundreds, if not thousands of hospitals, is not substantial manipulation. Also, it's antibodies binding to the surface of cells. And also radio labeling is not making something into advanced therapy. So you can see classification is not straightforward. And this is where regulatory science is so important. And also scientific understanding is important. So the regulatory agencies do have a lot of staff who actually former researchers who would understand these problems and would come up with a scientific solution to them. So how do you optimize then when you work as a researcher? And that could be a medicine one day. First, be aware. Then also define the product from early on and characterize it. So for example, if you're expressing a certain protein you're working on, collect as much information as possible. How does it behave also under stress conditions? That's what my group leader actually did with this gamma delta T cell receptor and also the antigen preparations. He characterized it by many different methods whenever he expressed it. That does not only enhance the quality of the publications, but also helps later on when this is then maybe upscaled and made as a medicinal product under industrial conditions for so-called comparability exercises. And that means nothing else than to show that the previous product version, small lab scale, is more or less, should we say, identical or highly similar to the industrial scale product. So any publication, any animal testing and so on that's been done before is not completely irrelevant, but actually very relevant. And you can see that this saves a lot of time, money, and effort later on. So the more information there is and also documented, the better it is. And if one already goes ahead to say, well, this is actually really going to medicines development, we'll soon going to have a spin out of our academic group, then it is good to already then look at manufacturing, define the process, how it's made, how it's expressed in bacteria, how you do it, and also check for problematic areas. So for example, certain growth factors, which might be of research grade or of animal origin, that could, the regulators will say they might be contaminated with known or unknown viruses. Antibiotics for selection, so selection markers, very widely used, but if it's ampicillin, for example, as or kanamycin, they are medically used and people may have an allergy. So it would be not so good if someone with kanamycin allergy could not use the product or get an allergy from getting or an hypersensitivity from the product and not be able to use kanamycin. It's used for eye drops, for example. It's very theoretical, extremely theoretical, but that's how regulators are. And the earlier you know, the better it is, because then one can think about alternatives or say it is an alternative, and this is what the risk assessment will actually look like. And then in the final part of the lecture, I'll briefly touch upon this now. The non-clinical models, not just tick box approach, what we did in the olden days with testing of pharmaceutical product candidates, but also select the models wisely and keep all documentations, all lab manuals, standard operating procedures, notes, and so on. The regulators want actually more than what is in a publication that I have seen several times, that researchers were actually saying, well, we've got all this in a publication. They want even more. Yeah, they do actually want more because they drill down to the detail. But don't be afraid of it. It is their job and it's normally all solvable. And don't be afraid to ask the regulators, Innovation Task Force, Innovation Office, scientific advice. That is a very important and also very often very positive experience for people. Now, in the last 10 minutes or so, I will just talk about non-clinical development. And what is non-clinical? Non-clinical is all studies that are not performed in humans. And that is not characterization assays, but that is animal studies, in vitro tests, which are functional tests, I should have added, ex vivo tests, and others. And there is a long list of requirements. I'm not going through all of these here, but you can see that there's a certain systematic to this. It's again in this Directive 2001/83 as amended. That's also the annex of the Arzneimittelgesetz, the German drug law. And some of these items here, like for example, carcinogenicity, is not relevant for some biological medicinal products. And the classical test for this is the so-called Ames test. You take bacteria, a suspension of bacteria, of a bacterial clone that is well characterized and the genome is sequenced. So you know you can identify every mutation coming. And as you know, bacteria have a short generation time. So you can see many, many, many generation doublings within a day or two. And then you put a toxic substance like an alkylating agent, is the classic, for example, chemotherapy, you put into the suspension, and then you measure how many DNA double-strand breaks and so on and mutations actually on average have occurred. And that is a measure of carcinogenicity. But if you put a monoclonal antibody into the suspension, nothing will happen unless the monoclonal antibody by just just by chance binds to anything on the bacteria and does something to the bacteria, but that's about it. So that test would be completely irrelevant. And this is where regulatory science says this is where the guidelines needed to be updated to say that carcinogenicity is not required for biological medicinal products. But if there is a mechanistic issue, like with embryonic stem cells, for example, which divide and divide and divide, or with gene therapies, which can integrate into vulnerable parts of the human genome, then what we want is tumorigenicity studies, but they have a different design. That's not a bacterial test. That's not an Ames test. That is a specifically tailored non-clinical study. And that is actually what I'm talking about when I say that the non-clinical development needs to be informed by regulatory science. And we have seen this in the hard way when Ulrich was still at the Paul-Ehrlich Institute at the time, and I was working with the monoclonal antibodies in 2006. It's long ago, but it's still relevant. It's still the 9/11 of medicines development, of biologics development. The TGenero case. TGenero was a small company and a small German company. They had one asset, TGN-1412, which was a monoclonal antibody that was tested in 6 healthy volunteers in a hospital in London. Then it after a few minutes, they all had shock-like symptoms and also had necrosis of the fingertips and toes and almost died. And what happened was a massive cytokine release syndrome, which led to a shock-like syndrome. It was also at the time in the German newspapers. And some of them also had multi-organ failure. So that was a really severe situation. So what happened? This was a monoclonal antibody against CD28, which would polyclonally activate T cells. So normally, alpha beta T cells would recognize the antigen presented in the MHC loop by antigen presenting cells, but as they swim around the body, so to speak, doing this with all cells, they would normally not want to be activated, only if there is a real problem, a danger signal, and that is co-stimulation. So that would be the binding of B7 upregulated antigen-presenting cells binding to CD28. Of course, it's much more complicated than this, but in this very simplistic view for understanding the mechanism of action, for TGN-1412 bound to a different loop of the CD28 molecule, which basically helps to circumvent the T cell receptor and activate polyclonally T cells. So why is that a good idea? This sounds actually already quite dangerous. And indeed, at the time, the agencies, it was the British agency because the clinical trial was applied for in Great Britain, and also the German agency, the Paul-Ehrlich Institute, at the time really scrutinized the dossier because that, of course, sounds like a medicine that has a certain risk. The idea behind this was as follows. Patients with chronic lymphatic leukemia of the B cell type, BCLL, often do not die from the leukemia. It's a very long-term disease, but they have a severe immunodeficiency because of non-functioning B cells, and also problems with how the immune system in general works. And the idea was to polyclonally activate T cells to a level that would be the normal activation level. You could try to titrate this with non-clinical and clinical data from the first in human clinical study so that they would be able to not only have a normal immune response, but also to kill the tumor. That was the idea behind it. And there were some data suggesting that regulatory T cells would be preferentially activated with certain doses, lower doses, I believe, and that would then help to generate regulatory T cells, which would then curb autoimmune diseases like multiple sclerosis, rheumatoid arthritis. So there is a typo in this. I just saw that, rheumatoid arthritis, of course. And the problem here is that a monoclonal antibody has to be shown to be safe in a relevant species. And that is defined as one in which the test material is pharmacologically active due to the expression of the receptor on epitope in case of monoclonal antibodies. And for this particular product, the sequence homology of CD28 in the cynomolgus monkey that was chosen as the model was actually 100% identical. So it was seen as a relevant model. And I'm not going through all the data now, but there were no signs of toxicity. Of course, no signs of toxicity already makes one think, is this actually something which is relevant? But of course, when the sequence homology is 100%, that's all you can ask for. And there was a moderate increase of some cytokines in the serum of single animals. So there was some pharmacological effect. And that was basically the tricky thing. With hindsight, everything was done according to the rules. So there was no deficiency, no oversight. But for this particular molecule, even the sequence homology of CD28 being 100% in the extracellular domain was not sufficient. And that led to a number of different considerations. I will not go through the details, but this is basically what regulatory science does. It does not say monoclonal antibodies from today are a banned product class, which is too dangerous to develop, and we cannot have products like anti-CD28 or anything activating in the immune system anymore from today. But it says, what can we actually learn from this? And how do we do this? How do we distill this into guidelines, into a new guideline, which actually also was prepared, a guideline for identifying and mitigating risk for first-in-human clinical studies. And we learned from this that it is not just the binding in the sequence homology, it is also agonistic and antagonistic downstream effects, and also functionality of the corresponding functional systems. And actually, Ulrich Kalinke and colleagues from the Paul-Ehrlich Institute also did a lot of research into this to find out what the problem actually was at the time. And regulators do also accept alternatives. There are some scenarios where there is no animal model, where the only relevant species is actually humans, like the classical tumor antigen EpCAM, for example, or CD3. CD3 is as it is human CD3 to my knowledge is only on human cells. So a mouse has a different CD3 structure, and in this case you can use model systems, transgenic animals, for example, or a surrogate antibody, as we call it. So a mouse anti-mouse CD3 antibody can give some valuable information on how an anti-human CD3 antibody would work. Of course, we know a lot about these particular ones, but if it is any other antibody where this applies, then of course that approach can be taken. And very importantly, data from a non-relevant species are not required. Regulators were very clear also at the time, which was actually quite good because it gave a lot of certainty to developers. Of course, for cell therapies, this is even worse because a relevant species is not just one epitope. There is multiple surface markers, receptors, integrins, and whatever on the cell surface of cells, and they secrete factors. So the microenvironment is acted upon, but also acts on the cell. So here, really, the only relevant species is humans, and this is why the non-clinical development for cell-based medicine products often looks much different and also very tailored in incompetent mice, for example, where you can do proof of concept, and the toxicology studies might sometimes be completely different. And what we've often seen before this case, the T. Gennaro case, was that companies would go very fast into clinical development. We have also seen cases where they did the non-clinical studies, well, more or less in parallel to the clinical, because they had to, because it was somewhere in a guideline. And then the non-clinical and the clinical assessors looked at this, and there was almost no consequence taken from non-clinical studies. Apparently at that time, it didn't matter too much because the clinical data were comprehensive. But of course, this is missing the point of what a non-clinical study actually does. And what we now see and want to see also is doing this first, gaining knowledge on the mechanism of action. So to have a real rationale for the non-clinical testing, not just because regulators are asking for it, to have it not reactive, but predictive. So what is going to happen in humans? What can we maybe not expect to see in animals? So a gap of knowledge and knowledge about that gap, is also a risk factor to take into consideration. And importantly, it helps defining the starting dose. And I will talk more about this non-clinical development and first-in-human clinical studies in one of the upcoming twin-core lectures. So to conclude, regulatory science evolves alongside pharmaceutical science and basic science, and is a very important discipline. Academic scientists ideally would be aware of the basics of drug development, since they might already be working on a future medicine. And I should add, should not be afraid of it. Non-clinical development is a regulatory requirement, but can be cleverly designed and is actually something which is really also useful to do. And regulatory authorities are not only gatekeepers, but also enablers. So one shouldn't be shy and actually consult them. So that was the end of my lecture.